Good day, biology. Today is November 5th. Today we are going to start. If you completed and submitted your targets, you've got your quiz to take. If you didn't finish your targets, finish your targets and make sure you submit those. You'll need your phone to take pictures and upload them. I got it. That's okay as long as you're not just texting and getting distracted on TikTok and Instagram and all that stuff. We've got Cooking with DNA Lab that we did yesterday with the Rice Krispie treats. I'll show you kind of review about that. We've got to paste in our targets. I'll tell you what pages those go on in just a little bit. They're going to go on to pages 92 and 93. And then our CK12s, we've got a couple of readings that are due by the end of Wednesday. So you've got one you could do tonight, one you could do tomorrow. Or if you got time, do them both tonight. I know a lot of you have tryouts and that sort of thing. So use decal to take care of that sort of stuff. You've got decal today. That's what you're going to watch right now, actually. I guess you're in decal now. So if you're in second period, right? So what type of mutation is seen below? What we have in the original sequence is just a C. So the other side would be G. It's just saving space by not showing both sides. On this one, instead, we have a T and an A, and that resulted in a different amino acid than what the beginning was. Now, this type of mutation may not always do that because some of our amino acids are coded for by multiple very similar sequences. But what do we call this? Is it insertion, deletion, or substitution? So go ahead, pause, take time to write all this down. All right, and then here is our answer, substitution. It's a substitution mutation. If you had an insertion or deletion, that's going to frame shift it, and that causes lots of different amino acid sequences. Sometimes changing one amino acid can make a minor difference. Sometimes it makes a giant difference. We're going to talk about how we get a big difference with the substitute mutation for sickle cell anemia. That's one thing that we're going to be doing our DNA gel electrophoresis with. I did want to talk about the DNA cooking with DNA lab. So I'm going to go forward right here. And I want you to see that we had three different versions of the same trait, all because the DNA code that you copied down was different at one place, all right? And that led to a different color. So this is just like different traits that we see in all sorts of organisms. One little mutation changes what that trait will look like, all right? So you are going to do a couple things. You've got your protein synthesis quiz um, if you turned in those targets. You've got biotechnology and genetic disorder readings. Those are kind of the last thing that's to do. Really important. I'm going to put this at top. Submit cooking with DNA analysis. This is going to be a summative. I'm going to grade it as a summative. So make sure you look closely. You answer your questions thoroughly using complete sentences. All right. We've got this all completed. You're going to answer these in complete sentences and you're going to write a conclusion. What did you learn? Read this carefully. It says construct explanation based on evidence for how structure of DNA determines the structure of proteins. Right? This is kind of summarizing what we did in the lab. Talk about these guys. Okay, that's going to help you. This, if you talk about these guys, that will pretty much give us your answering the question correctly. Relate it to what we did with Rice Krispies. You can type on here, you can write on a separate piece of paper. You're going to need to take a picture of everything and upload it to here. I right now am going to change the due date and make this due Friday. Okay, so it's due Friday. Due Friday the 8th. Okay. Now, you should be able to get most of this done today, but I want you to take your time. Uh, there we go. What else did I have to say? I just want to make sure I got over it. Cooking with DNA, got that. Paste in targets. Those targets, again, are going to go on to page 92 and 93 in your notebook. That's the wrong class. There we go. 92 and 93 in your notebook. Yep. Guiding questions and vocab, targets 3-3. Your protein synthesis lab can get pasted all on page 91 as a super fancy flip, that sort of thing. All right, that should be it. I'll be down the hall if you have a question, and you guys have a great day. Thank you.